This is the Mass Spec Pen 2.0, revolutionizing histopathology.
Many say that this pen is mightier than a sword, and that saying holds true, especially when you take in consideration how this pen is able to make biopsy a thing of the past.
The Mass Spec Pen 2.0 can quickly detect cancerous lymphocytes and distinguish them from normal cells by touch. Our innovation focuses on expanding the biochemical analysis of lymphomas to detect certain or specific types of lymphomas and subsequently administer nanoparticles to destroy these cells.
What are we looking for?
We're looking for B-cell lymphoma. So basically, I'm just going to take the pen and touch it with the tissue. So, can you read it and see if we have cancer in here?
It is affirmative. This patient does have
So
cancer.
then we'll just administer the nanoparticles now. So I administer the nanoparticles.
Okay.
So basically the end of the procedure. Quicker than a biopsy.
Job well done. Job well done.
So, when the pen comes in contact with live tissue, a drop of water extracts biomolecules from it. The water containing the molecules is drawn up the mass spectrometer. Next, the mass spectrometer analyzes the molecular composition, detecting the type of cancer cells. A compartment in the pen houses different types of nanoparticles. Coordinating with the mass spectrometer, the compartment releases nanoparticles that will be fed to the cancerous tissue. In the case of B-cell lymphoma, the nanoparticles mimic HDL, high-density lipoprotein, particles in size, shape, and surface anatomy. These three factors help the compartment process the information received from the mass spectrometer, almost like a blueprint for attack. B-cell lymphoma needs a constant supply of HDL to survive, and the nanoparticle disguised as HDL enters the cell by way of endocytosis. Some of those cancer cells, they're not there anymore, they're not present, so they've basically died because of the nanoparticles.
Go. I think I'm sure.
Essentially, our innovation seeks to minimize the time between diagnosis and treatment from the or to the pathology lab.